Hello, I'm Dorina Landa, and I'm the Executive Director of Advancement at the University of Toronto's Temerty Faculty of Medicine. I'd like to begin today's event by acknowledging the land on which the University of Toronto operates. For thousands of years, it's been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still home to many Indigenous people from across Turtle Island, and we are truly grateful to have the opportunity to work on this land. And it gives me great pleasure to welcome you today to this latest installment in our Temerty Medicine Talk series. Today's discussion will focus on new frontiers in our understanding of ALS, Alzheimer's, and Parkinson's, all of which are priority areas of focus at U of T's Tan Center for Research in Neurodegenerative Diseases. I hope you enjoyed today's discussion, and I very much look forward to hosting more Temerty Medicine Talks in the future. I'd now like to turn it over to Dr. Patricia Houston, Vice Dean of Medical Education, and currently our Acting Dean of the Temerty Faculty of Medicine.
Thank you, Dorina. It is my sincere pleasure to welcome you all to this first Temerty Medicine Talk of our new academic year. This series was established as one of the ways that we can share some of our remarkable work underway in our faculty and in our partner hospitals. It allows us to showcase our ongoing commitment to equity, discovery, and collaboration, the three pillars of Temerty Faculty of Medicine's strategic plan. I'm excited to also share that these accomplishments are not going unnoticed. Just 3 weeks ago, we learned the University of Toronto is now ranked 5th in the world by Times Higher Education in the clinical and health category, up 1 spot since 2021. This is powerful recognition of Temerty Medicine's research, education, and patient care, and confirms our position among the world's best faculties of medicine. The Tan Center scientists and clinicians you'll hear from today are a perfect example of why we have earned this international recognition. They are tackling medicine's big questions, working to develop innovative solutions that will advance health and healthcare for our patients, their families, and our communities. I'm also pleased to be welcoming back André Picard, the award-winning health columnist for the Globe and Mail, who will help guide our conversation today. André, thank you for being with us yet again this year. And I'm going to turn it over to you now to introduce our topic and our panelists. Thank you again.
Great. So thank you very much, Dr. Houston, for that kind introduction. As you know, the speaker bios are in the chat. I'm not going to go through their long bios because we would not have any time for chatting because they're a very distinguished group. So I'll just again introduce them very briefly. Dr. Graham Collingridge, Lorraine Callia, and Janice Robertson. Thank you for joining us today for this discussion on neurodegenerative disease. And Graham, I wondered if I could start with you just to set the context, set the base here. Tell us a little bit, what is neurodegenerative disease, and what do conditions like Alzheimer and Parkinson and ALS, what do they have in common that puts them all in this same group?
Right. Well, thank you, André. So they are, as you point out, a collection of diseases which ultimately lead to destruction of nerve cells in the brain. But depending on what part of the brain that affects, they manifest as motor disorders, such as in Parkinson's disease or ALS, or cognitive dysfunction, as we see in Alzheimer's disease. So they are distinct in terms of how they impact the patient, but there are a lot of common underlying mechanisms. And what we do at the TANS, because we have researchers working in all these different disease areas, is we look for common underlying mechanisms, things that link, for example, Parkinson's disease, ALS, and Alzheimer's disease together.
And this, the TANS, this is something it did many years ago. It started looking at the common mechanisms. These used to be all live in different worlds, right?
Absolutely. I mean, the TANS, when it was founded, and this happened long before I was here, but under the previous directorship of Peter St. George Hislop, they had the idea that they would bring together people from different specialities, different disciplines, and put them under the same roof to work together on these common problems. And that was unique in Canada at the time. Now the approach to neurodegenerative disorders is an international effort involving many, many different techniques and many collaborations around the world.
Great, and we'll talk a little later about this importance of collaboration, but I wanted to bring Janice in here. We have a lot of questions that have come in ahead of time, and a lot of them are about genetics. People are fascinated by genetics, genomics, and people have a sense that all these things are kind of inevitable, that we're doomed by our genes. Is that a correct perception that the public has?
I think certainly, as we've discovered genes, it gives us a handle on understanding the diseases. And certainly, if you carry certain gene mutations, for example, in ALS, the disease that I work on, if you carry a mutation in a gene called superoxide dismutase 1, you will get ALS at some stage in your life. So there are certainly diseases that we call 100% penetrant, meaning that if you carry a gene mutation in a particular gene, you will get the disease. But there's also, variabilities on whether or not you will get the disease. Maybe they're dependent on lifestyle choices, which we don't really know a great deal about. But yes, largely, 5 to 10% of ALS cases and Parkinson's disease are genetic. And we know that through the family histories, that if you carry these genes, then you will get the disease.
5 to 10%, that's not most. That's a very little number. People don't necessarily have the right perception. These things are random. Is that a way to put it?
Well, 5 to 10% are family history. 90%, I'm talking specifically here about ALS, is sporadic, meaning that we don't really have a clue what causes the disease, whether there are maybe some deficiencies in genes that we don't know about yet that may contribute to you manifesting with ALS. That's still under intensive investigation. So, most focus has been on the genetic forms of the disease because this is what we know is definitely causing the disease, and it gives us a handle on trying to understand the mechanisms that are causing the disease. So maybe that's the perception that the public has, but yeah, 90% we really don't know what's causing it.
Okay. Now, Lorraine, you work as a researcher, but also a clinician. A lot of these things are scary. Brain diseases are scary to people. How do you deal with that when people come in, say, with a dementia diagnosis, ALS? How do you address their fears? Because the media perception of this is, boy, you're in big trouble. It's not going to be a nice life.
Yeah, that's a great question. As you mentioned, I am a neurologist, and most patients I see have Parkinson's disease, but can also have much more progressive neurological conditions that also fall under the umbrella of neurodegeneration, such as rare diseases such as multiple system atrophy and progressive supranuclear palsy, which sometimes can mimic Parkinson's disease. And I think what the challenges are, one aspect of Parkinson's disease that I will always point out to patients that I see with this diagnosis is, of the neurodegenerative conditions, the silver lining for this diagnosis is that we actually have some very good therapies. We call them symptomatic therapies because they treat the symptoms. So if somebody has a tremor or if somebody is slow to move, we can replace a brain chemical called dopamine in their brain and actually replace what is actually missing from the brain cells that are dying, and can help things like tremor and mobility. What we don't have is treatments that can actually stop that brain cell death that Graham mentioned. And that really is, I think, an area that we're all actively very much working on at the TANS to try and find that kind of therapy for all of the neurodegenerative conditions. But for Parkinson's disease, people can have a very substantial benefit from the symptomatic therapies. We also have surgical therapy, so deep brain stimulation, which work in a symptomatic way in terms of managing symptoms, but not changing the progression of the disease. And those I find are silver linings in this condition, and people can actually have many, many years of good functioning. And so that is the one positive piece that I try and emphasize with people that I see in the clinic. And I think the second really positive piece is that we're not sitting on our thumbs. We're working really hard to try and understand these diseases, and we are making progress. And I think as we understand why it is that these brain cells die in each of these diseases, it leads us down that path as to find actual new treatments. I have I like to think relatively early in my career, although time goes by quite quickly, but I do think that we are far further ahead than we were 10 years ago, or even when I was a trainee 15 years ago and first seeing these patients. And so I have hope, and I hope that that gives hope to the people who I see in clinic.
Great. And Graham, if I could come back, I want to just build on what Lorraine said, that we've come a long way in the last 10 years. Can you, I'm sure you share that view. Can you give us a sense of how things have changed, especially in your area, in the dementia area that people are very interested in? Has there been a lot of positive change?
Yes, absolutely. We have learned so much more about the basic biology over the last 10 years. In the terms of dementia, I mean, with Alzheimer's disease, it's very similar to, as Janice explained, for ALS, that there is a small population which is determined purely by genetics. We know that if people have mutations in certain genes, in this case, it's the amyloid precursor protein or the presenilin genes, then they're going to get Alzheimer's disease. But in the majority of cases, that's not clear. There may be a change in a single protein which will increase the probability of getting Alzheimer's disease by a tiny percentage, and it will be an interaction with lifestyle choices. So we know now that it's much more complex than we originally thought, but we also have an insight into the underlying mechanisms that all of these genetic changes are converging onto a single set of molecular changes which control the communication between the nerve cells. So in the human brain, we have about 100 billion neurons, which are the powerhouse of the brain, the nerve cells which communicate. And they communicate with one another through connections called synapses, of which we have about 100 trillion. And that's how we store our information. And that's where all the cognitive processing goes on. And what's become clear over the last 10 years is that what causes the dementia initially is a loss of these synapses, because the basic mechanisms which control how they function breaks down. And as we're understanding that, then we can start to target treatments specifically to correct those errors. And there's a couple of good examples in Alzheimer's disease already.
And you mentioned we've realized it's more complex, but we have more knowledge. Who's winning that battle? Are we getting there and making progress?
We're definitely making progress. Yes, we're learning it's more complex, but that's the reality of the situation we face. I mean, if dementia was simple, we would have solved it by now. I mean, we're dealing with the most complex process that we know about in the known universe, the human brain. And it's very, very complex, involving an interplay of lots and lots of different molecules and the external environment. And really, one of the things we have learned a lot over the last few years is how much we can influence the progression of dementia through lifestyle changes, because it very much is an interaction between our genetics, which we can't change readily, and our lifestyle changes, which we can change. And in fact, there was a very influential publication in The Lancet last year, which identified that 40% of dementia is potentially modifiable by lifestyle changes. So only 60% is intrinsic. So each one might only have a small effect. So for example, early life education can protect against dementia later in life. But giving up smoking can reduce the risk of dementia. Excessive drinking, giving up excessive drinking can reduce the risk of dementia. Combating depression can reduce the risk of depression. So we add these all up, it makes a significant difference. So there's a lot that we can actually do ourselves. And indeed, just recently, a couple of years ago in the US, there was a study to show that actually in people who are really taking care of their health, the incidence of Alzheimer's disease is decreasing in that population. And so there is a lot of hope.
Great,
definitely.
Great. We don't hear that often in my field, so that's great to hear that word, and we'll come back to that. Janice, I want to ask you the same question. Your specialty in the ALS field, that's one of the most frightening diseases imaginable to people. Is there more hope there? Is there more progress, better treatments? Have things changed in recent years?
Yeah, things have changed dramatically in recent years. And one particular story is the gene that I had mentioned earlier, superoxide dismutase 1. The mutations for that gene were found in 1993. And through the following years, when ALS was not such a well-studied disease, it was often, when you say you work on ALS, people say, oh, is that the same as MS? It was not a well-known disease. And, labs worked away in trying to figure out how these mutations in that gene, it was the only tractable piece of evidence that we had what caused the disease. And our group and others had established that the mutations within that gene are causing the protein it produces to misfold. And it adopts an abnormal shape. And it's through this abnormal shape that precipitates the onset of the disease. And so in 2014, we had this incredible event called the Ice Bucket Challenge. And through that had led to therapeutics targeting this misfolded form of SOD1. And this has led recently to a drug called torfacid, which is an antisense oligonucleotide. And by all measures, it's proving to be highly effective in the phase 3 clinical trials. And I think there's going to be announcements coming very soon about how hopeful we are about that particular treatment, which is an antisense which actually targets the mutant gene and downregulates that mutant gene. So the original discovery was made in 1993, and it's a really long time. And of course, people with ALS have a very rapid progression of disease typically. 50% of people die within 18 months. Most people die within 5 years, but there are obviously people who can live longer than that, especially with treatments in the clinic in that, and care, and nutrition, etc. People can live longer. But now there are far more people working on ALS because of things like the Ice Bucket Challenge, with 100 companies right now looking at therapeutic targets for the treatment of ALS. So there's great optimism in the field and for the clinics in general. But for the patients, of course, it's not fast enough, especially for a disease like ALS that is so rapid. So one of the main criteria right now is, if a new drug comes to Canada and it has to pass through Health Canada for approval, it's not going fast enough for patients who really want to have a chance at trying new treatments. But in Canada right now, I think there's about 15 clinical trials ongoing in Canada right now, and about 100 going on around the world. So yeah, there's a lot of optimism. And this new one for SOD1, this antisense oligonucleotide treatment, that came about through all of the science and research that went into understanding how these mutant genes actually elicit the disease.
And you mentioned, this community, the ALS community, is very active. The Ice Bucket Challenge. I recently saw a great movie about Addie Barkan, the activist in the US. You're really dependent on patients, and some of them, as you mentioned, have a very short lifespan ahead of them, but they still dedicate so much of their lives to research. Talk to me about how important that patient input is.
They're an incredible community. They're obviously fighting for their lives, the families. And one of the things that we have learned in the past several years is that ALS, like Alzheimer's probably and Parkinson's disease, is that it's not a single disease, that each person is an individual, and we need to learn more about these individuals. And through this, I'm part of an initiative that's gone across Canada called CAPTURE, which is the comprehensive analysis platform for the understanding, remedy, and elimination of ALS. So it's called Capture ALS. And basically what this involves is clinics across Canada uniting together to completely analyze individuals, people, in terms of their genetics, their various clinical measurements, their age, their backgrounds, so that we can really try to identify subpopulations so that, for example, the SOD1 that's targeting that specific gene may not work in all patients and likely will not. So we need to be able to identify the right treatments so that we can give them to the right individuals. And I think we're going to see much, much better outcomes in clinical trials through that initiative.
Great. Now, Lorraine, to build on this, patients are much more active today. They're much more engaged in their care and their treatment, but that's not always easy for a clinician. I'm wondering, how do you deal with the people who are guided by Dr. Google and Dr. Twitter, don't always necessarily have the best science? How do you deal with that?
Yeah, so that's a great question, and it's something that we deal with every single day. I think it's wonderful. I think it's amazing that people are so interested in their health and taking initiative to try and learn more about their disease. And I embrace that and completely support that. And at the same time, I try and be a resource for patients to try and understand how we want to make sure that they have opportunities, that they are able to explore, different options, but at the same time do it in a safe and scientifically sound way. And so patients come to me often with lots of ideas, and I think the first step is just to be open-minded, because there is a huge amount of information out there, and sometimes people come with anecdotes or findings or readings that they've had that I think do require us to kind of look into a little bit more. But we always have a conversation about risk versus benefit. What's actually studied, what do we know what the risks are around things, and if we don't know what the risks are, how safe should they be in terms of trying or exploring different things. And then there's also that risk that I often talk about, which is the risk to their wallet. So we always have to be cognizant of people who are out there trying to sell the kind of snake oil to people. And we have a balanced discussion. If I need to go back and do more reading and see if there's actually some evidence behind what patients are discussing and whether or not there might be a trial that they can participate in related to what they're interested in, we explore that. A very common example is somebody coming into my clinic interested about CBD oil. There aren't a lot of very good studies in Parkinson's disease using CBD oil. Patients will try it, and I'll share anecdotes that I've had with other patients, but I also will direct them to clinical trials that we do have ongoing if they're interested in participating and adding to that knowledge. So for example, we have an ongoing clinical trial by my colleague Susan Fox in our clinic looking at CBD oil for the use of pain in Parkinson's disease. And I think going back to engaging patients, I think this is another way to really engage patients in research, that patients these days really are trying to take hold of their kind of autonomy. And with that, I think it's wonderful because it will really help in terms of pushing our understanding of disease ahead and allowing us to really have them engage in research in ways that include them both being subjects in research trials, but also providing input as to what is important to them in terms of what we should be researching.
And again, in your field, there's some high profile people. Michael J. Fox has really brought knowledge about Parkinson and also lots of research money. That's important too, those voices.
Absolutely. And, I'd say Michael J. Fox would be analogous to the ice bucket challenge that Janice talked about for ALS. I mean, Michael J. Fox has not only put a face on Parkinson's disease, but really put his money where his mouth is and has generated not just funding for research, but a community of researchers and a direction for research to go, and has changed, I think, the face of Parkinson's disease research for decades to come. And I think the importance of that is really priceless.
Now, Graham, while we're talking about research, I wonder if you could talk a bit about the importance of fundamental research. We in the media, we love the new drugs, we love the new treatments, but it starts in the lab with test tubes and rats and all that. That stuff's important.
Oh, absolutely. I mean, we wouldn't be able to do the clinical trials if we didn't do the basic research first. I mean, a good example of this, and I know the COVID analogy is used extensively, but it really is a very good example that Pfizer was able to produce the vaccine very quickly because of the work done by the biotech companies, and they were able to do their work very quickly because there was 10 years of fundamental research into the principle of mRNA vaccines. And it's the same with what we do. If we want to develop a new drug, if somebody wants to develop a new drug, if a Pfizer or Johnson & Johnson are going to do that, they need a large amount of basic information to build upon. And that information almost invariably starts in universities and research institutes, such as the TANs. So it's really important to invest at the fundamental basic research level.
And as Janice mentioned, she mentioned an example of in 1993 research began and the drug is now a reality. So can you talk about the research part? What about the sharing? This isn't, you just don't collaborate in the TANs, you collaborate around the world now on these diseases.
Absolutely. So we're a very collaborative research institute ourselves, and we're embedded within a UHN hospital. So we collaborate with the Krembil Discovery Tower researchers as well, who are neuroscientists, many of whom have similar interests. But then within Toronto, with this fantastic network of dementia researchers through what's called the Toronto Dementia Research Alliance, which brings together several research intensive hospitals, such as the UHN, such as CAMH, with the University of Toronto, So we've got a huge network here in Toronto, and then that extends beyond within Canada, where, as Janice has already mentioned, you get lots of networks of people working together. Within Ontario, we have a massive emphasis on dementia too. And then this goes beyond Canada, because the problems that we confront, Alzheimer's disease, Parkinson's disease, ALS, etc., are worldwide problems. And the solutions are going to come through collaboration between scientists here in Canada, the US, Asia, Europe, etc., etc. So it makes sense for it to be collaborative. It's the way that we can drive science forward the most efficiently and the most effectively. And that's what everybody wants to achieve.
Great. And I want to, I guess, I come back to Lorraine on that. What's the importance of you knowing things about ALS and Alzheimer's? Does that make a difference, or do you just have to stay really narrowly focused on your area, or does that collaboration across different neurodegenerative diseases, does that matter to you?
It's super important. I mean, we know that there are general principles that are common across the diseases. You know, I could tell you also anecdotally that there are patients who we like to put people in categories, but there are also patients who will span these different disease categories, which I think is just a demonstration of how there is a lot of overlap between these diseases. And, you know, I'll take an example from Janice's world, but there was a time when people felt that ALS was its own disease and this entity called frontotemporal dementia was its own disease. And we now know that it's a spectrum. And, you know, there's similar types of scenarios in the Parkinson's world, but you can kind of describe that across all neurodegenerative conditions. And I think getting back to Graham's point about the complexity, one of, I think, the aspects of the complexity of these neurodegenerative diseases is we kind of like to make things simple, to put people in separate buckets. But we now know that tons of pathology that you might see in an Alzheimer's patient at autopsy, you'll also see in a Parkinson's disease patient at autopsy. And so that just speaks to the fact that there's a huge amount of overlap in these conditions. And, you know, our hope is that if we kind of make breakthroughs in one area, that might be translatable to another neurodegenerative disease because they share these common mechanisms at the cellular and molecular level. And I think that will turn out to be true. And it is a kind of complex notion, because I'd also argue that one of the great things and the aspects that have come out of the complexity of these diseases is that we've also, as Janice already mentioned, appreciate that each person is individual too. And so there's this consideration that we also have to make in terms of this concept of precision medicine, which many people have heard of in the context of cancer, that I think in the very near future will also start to have to be applied to neurodegenerative diseases. And so there's this overlap of all of the diseases, but then also this very individualized approach to each patient that I think is going to start to push the field forward.
And Janice, I wondered if you could build on that. You work with a disease that's relatively rare. Is that the future, really personalized, figuring out the genome of that person or whatever and adapting the treatment?
I'd just like to just address the point that it is a rare disease, but the reason that it appears rare is because people die so rapidly. So I just like to, you know, put that in perspective, that if we had better treatments, you'd see a much larger ALS population. It's just that they die really quickly. I think that it really speaks to the strength of the TANs, that we have their researchers and clinicians working on these different diseases all under the same roof. And so we are able to, we're all in our own networks, in our own kind of disease worlds, but we're interconnected at the TANs. So I learned from Graham, I learned from Lorraine, I learned from the other members of the team, and we keep each other abreast of everything that's going on in the field in terms of genetics. And so, you know, as Lorraine was saying, this overlap, this complexity of these diseases, you know, ALS with another disease called frontotemporal dementia, it was discovered, it was always kind of known at the clinical level that there was an overlap, but it was established in 2011 with the finding of a gene called C9orf72. And that gene has a piece of DNA that is expanded in length, and that is called a nucleotide expansion disease. Now, when we think of nucleotide expansion diseases, we're thinking of things like Huntington's disease and Fragile X syndrome and things like that. And we never knew that ALS was going to take us into that world. as well in terms of all the work that's been done in those diseases. We have now been able to feed into in terms of trying to understand ALS and frontotemporal dementia. So it's a large continuum, and all of us are working with groups in Canada and across the world in these vast networks. And that's the reason I really enjoy working at the TANS, is that it all feeds back into this one center where we can all work together to try to figure out what's going on in these diseases.
Okay. And Graham, on that point, we've been talking about these are spectrums. These are very, there's I don't know how many types of dementia. There are many types. Can you talk about, there's a lot of interest in the questions from the audience about the challenges of diagnosis. How do you figure out when someone has dementia, what kind, and how important is that?
Yeah, I mean, that's obviously very important. It's also very challenging, but we are developing new cognitive tests, which are more sophisticated, and also biomarkers, particularly imaging, to look at, in the case of Alzheimer's disease, the deposits of beta amyloid and also the tau protein. So we are able to start to stratify patients, because it is going to be important to be able to apply, as Lorraine said, precision medicine approaches. So that's something which is definitely going to become more and more common in the future, though it comes with a massive cost implication, because these scans are not cheap. And so we need to find ways of finding more effective ways of providing the stratification. But I mean, probably this is going to be answered better by Lorraine and her clinical expertise in terms of biomarkers. Yeah, Lorraine, if you want to jump in, yeah.
Yeah, sure. Biomarkers is a huge area. And again, this is a shared area, I'd say, across all of these neurodegenerative diseases. Historically, these diseases are definitively diagnosed based on what the patient looked like in life, but also what they look like at autopsy. And clearly, that's not an ideal scenario. We want to be able to diagnose patients in life, especially when we start having special therapies for each of them. So, I mean, the biomarker area is a really huge area that's, I think, moving very, very fast. Imaging is important, as Graham already mentioned, but perhaps gonna come with aspects of practicality. I have to say, in my own area in Parkinson's disease, and an exciting area that's, again, is being used across neurodegenerative diseases, and actually was borrowed from a rare, very, very aggressive neurodegenerative disease called prion disease, or some people will know it as Creutzfeldt-Jakob disease or mad cow disease in some scenarios. But there are some tests that we've borrowed from that area, where you can actually take the spinal fluid of patients and be able to detect in an indirect way these proteins that aggregate or clump up abnormally. This kind of method is actually being used for diagnostics for Creutzfeldt-Jakob disease, and is very much probably going to end up being a helpful diagnostic test that we could do with patients in clinic early in their disease for Parkinson's disease. And similarly, people are trying to develop similar tests for related neurodegenerative disease. But I think another good example of how learning from one neurodegenerative disease can actually quickly translate to changes for another neurodegenerative disease with important implications, and in this case, diagnostics. We're not quite there yet, so I'd say one of the challenges and probably frustrating aspects for patients when they come to a clinic is our inability to say with 100%, this is exactly what you have, on that first day that we see them. But again, I think we're getting closer to the point where we will be able to send them for an imaging test plus a CSF or a spinal tap, and maybe an additional blood test, and with that be able to say with far more certainty than we've had in the past to say, yes, you have that disease.
And if I could jump in there as well, I think Alzheimer's disease, with the measuring tau and A beta in blood and serum, has proven that these changes can occur well before the onset of the disease. And in fact, I think through studies of patients with Alzheimer's disease carrying, say, presenilin mutations, they know that the disease has actually started 25 years before it manifests in clinical symptoms. And so, A beta and tau are I think, are actually going to be moved into the clinic as reliable tests for Alzheimer's disease. I'm not sure if you're using that in your clinic, Lorraine.
We're not using it in our clinic now, no, but we don't typically see Alzheimer's disease patients.
Yeah, so I know that within our center, we have Carmela Tartaglia, who's actually developing biomarker assays, and through studying progressive supranuclear palsy, frontotemporal dementia, and interestingly, the concussion syndrome patients as well, looking for tau biomarkers within serum. Mainly, I think also to act as a surrogate measure for therapeutics as well. If you see elevated levels in a patient and you give them a treatment, then you would like to see decreased levels of certain proteins within these biological fluids. And that's something that this CAPTURE program that's funded by Brain Canada and ALS Canada is set out to do for ALS patients, is to identify these biomarkers so that then not only, as you say, when they come into the clinic, you're never sure, 100% sure if that's the disease that they have. And it can take 1 year, 2 years sometimes for ALS patients to get a definitive diagnosis. So to have a blood test or some other simpler test is critical so that any potential therapies could be delivered earlier on in the disease course when it's probably more likely that you're going to have some kind of benefit.
Yeah, and to your point, Janice, I mean, we don't typically use it. We don't typically have patients who come in with Alzheimer's disease as the question, but that being said, getting back to the overlap piece, right? There are definitely patients who will come with Parkinson's disease and Alzheimer's disease, and similar to what you said, when and if we have treatments that are going to target tau and A beta, there are going to be patients who have Parkinson's disease, Alzheimer's disease overlap, and we're going to want to know those patients so that we can actually treat them with an A beta or a tau specific therapy.
Mm-hmm. Great. I'm sorry I dropped off there, the joys of technology, and I'm back. So I'm sorry I missed a little bit of that last answer. I want to try and, we don't have lots of time left, but I want to try and answer some of the other audience questions that have come in. A lot of questions about diet. I don't know why. I haven't studied your bios enough to know who can tackle this one, but I don't know, Graham, if you mentioned how to prevent Alzheimer's. Does diet come into any of these neurodegenerative diseases? Lots of interest in that.
So a general rule is a healthy heart leads to a healthy brain. So things which are good for the heart are good for the brain. And it's probably for two reasons. One is because it improves cardiovascular function, which is good for the first reason. But secondly, it's the same processes that are good for our heart cells, our heart connections. It's the same for our neuronal connections. So as a general rule, a healthy, balanced diet will help reduce the risk and the progression of dementia. And I'd imagine, though I'm not an expert, that the same applies to the other neurodegenerative conditions too.
Well, I think for ALS, people can lose weight despite having, a high-fat diet. And so nutrition is incredibly important for people with ALS. And then there's another component that we haven't mentioned is the increasing interest in the microbiome and what bacteria do you have in your gut. And this gut-brain axis is incredibly interesting, and I think we're going to see a lot coming from that area in the years ahead.
Yeah, very good area.
Yeah, for sure.
So
I tell my patients the same thing when they come in. Healthy diet is the way to go. There is some evidence actually for the Mediterranean diet to be beneficial in Parkinson's disease. And as Janice mentioned, I think that this gut-brain axis is going to be, we're going to see a lot there. Parkinson's disease is probably taking the forefront of that right now because we know that many symptoms related to the gut actually precede the symptoms that would lead somebody to get diagnosed with Parkinson's disease. So constipation is remarkably common. And there is some pathological studies to suggest that the protein that we see that is abnormally aggregating in the brain in Parkinson's disease may actually start to do so in the gut at an earlier stage. And there's a lot of interest to know how the bacteria that normally live in our gut and the inflammation that results in our gut, how that might trigger the disease.
And
So-
you're all speaking generally about diet. I'm going to assume none of you have a magic bowl of blueberries you eat every day to cure all or anything like that. So, okay, I see the nodding. Lorena, while I have you on, ask you another question we have that's come in about you're working on a trial with focused ultrasound that's really promising.
Yeah, so it's an early trial, and maybe I'll use it as an example to touch on a couple of things that we've already discussed, just to re-highlight them. So I think the first really important part about this trial is that it's remarkably collaborative and interdisciplinary. It wouldn't be a trial that one could do in, that I would be able to sit and do all by myself in my isolated space. It required collaboration across the entire city. So this is a collaboration done together at UHN with Sunnybrook Health Sciences Center, with Nir Lipsman and the group up there. It's interdisciplinary, and that involves myself as a neurologist. It involves neurosurgeons such as Nir Lipsman and Sunil Kalia, who happens to be my husband, as well as the basic scientists who work on the actual technology of being able to use this focused ultrasound to what I'll explain to you what it does in a moment. But with that, I also want to point out that the basis of what this entire clinical trial is entirely depended on fundamental science. So it depended on physicists trying to use focused ultrasound, but it also required us to understand what molecules we think are really important in terms of causing neurodegeneration and Parkinson's disease. And so what the trial actually is using focused ultrasound to temporarily open up the blood-brain barrier, which is a naturally occurring barrier we all have that protects our brain because it's so important from toxins, but as a result also prevents drugs that maybe we want to get into the brain to get there. And so Clairville Heinonen has worked on this technology for very many years now and has a huge and very effective team up at Sunnybrook Health Sciences Center, where from the lab all the way to the human being, they've been able to develop a technology where little microbubbles are injected intravenously, and ultrasound is used to move around those microbubbles. And by doing so, it temporarily opens up this barrier, which is basically a layer of cells, and just opens up the space between the cells to allow things to go through temporarily. What we did, so Heinonen and Nir Lipsman already have used this technology to open up the blood-brain barrier in Alzheimer's disease and ALS. And so I'll use this again as a highlight of how we can use this across neurodegenerative diseases, and had shown it to be safe in those conditions. So when we started this collaboration with Nir Lipsman, our goal was to try and actually use this to deliver an agent. So we're doing that for the first time in Parkinson's disease. And the agent that we're delivering is an enzyme called glucocerebrosidase, which at the bench we have, and from genetics, we knew that this enzyme is deficient or doesn't work well in many people with Parkinson's disease. And so I'll bring up again precision medicine. So we've used a precision medicine approach in which we've taken people who have mutations in that gene that are responsible for making this enzyme. And we are trying to test in them, first off, the safety, which is the most important part. And that's the only place that we're at right now, is testing whether or not it's safe to use this technology and deliver this enzyme in patients with this type of Parkinson's disease. I hope that wasn't too long-winded.
No, it was a good detailed explanation that really touched on everything we talked about today: the cooperation, the mixture of disease sharing, the whole range of genomics to technology to everything. So it's a good example. But unfortunately, we're almost out of time. So I want to ask you each something to take home. I know you've all been very hopeful, but I asked you about the last 10 years. So I want to send people home. What's making you really hopeful about the next 10 years? What's that life will be better for patients with neurodegenerative diseases, or maybe even that we'll have fewer? Maybe Janice, you can kick us off. What's giving you hope?
Well, I'm really excited about the antisense oligonucleotide therapies. I'm also really happy that we've got to the stage now that we're really, it's called deep phenotyping people with these diseases, really trying to figure out what they have on an individual basis, because I think that in prior years, when clinical trials were run, patients that were enrolled in trials were not necessarily getting the treatment that might have had benefit for them. So I'm very excited that we're going to really nail down what treatments are best for the right patient group. And I think there's a lot of optimism in the community, and with a number of companies, 100 companies now, working on therapies for ALS, that we're really going to see some big changes in the next few years, but not fast enough.
No, it's never fast enough, but
never fast enough.
Still hopeful. Great.
Yeah, very, very hopeful.
Graham, dementia is an area that a lot of people have interest in. What's giving you hope that things will get better in the decade to come?
Well, for the same reasons as Janice has just said, we're getting a much better understanding of the need to stratify patients. I'm particularly excited by the increase in knowledge under the basic mechanisms that are underlying dementias. So I think on this much better knowledge, we will be able to develop more effective therapies. So I think this is a really, really good time to be in this area. We still need to understand more, of course. I mean, it's such a complicated system. We need a lot more basic fundamental research, but we're in a position that we can really start to effectively and target the sorts of treatments that should provide much better outcomes for our patients.
Great, thank you. Sorry, I just muted myself. I had some noise in the background there. Lorraine, I want to give you a chance to give us your hopeful take home.
Yeah. And actually, so I'm just going to repeat some of the same. My hopeful message is exactly the same, that we know far more now, and that, you know, people talk about failures that we've had in neurodegenerative disease in terms of trying to find treatments. And I think now we realize they're not truly failures. It's just we were taking strategies, assuming everybody was exactly the same, and making the assumption that this one therapy was going to work for everybody. So I put a positive spin, and I actually don't think that it was failures. I think it's just we needed more knowledge. And I really do think we're far closer to having the knowledge that we need, so that when we're testing specific therapies, or we have therapies that we think are going to be helpful for people, we're going to test them in the right person. And that's really going to tell us whether or not a therapy works or not. And then we can talk about things that really work or really don't work. But I think all of what we thought were failures in the past, I think we're going to be able to explain that and definitely learn from that. And so that's what keeps me optimistic going forward.
Great. Well, thank you all for ending on an optimistic note. And this area of great interest to people. I want to thank our speakers, Graham Collingridge, Lorraine Kalia, and Janice Robertson. To the audience, I urge you to please take a minute to fill out our survey. There's a link in the chat, and there's also a QR code on the screen if that's easier for you. And again, thank you all for being there in such large numbers, and thanks to our panelists. And we'll see you at the next Temerty Medicine Talk.
Thank you, André.
Thank you.